I'm going to give you today's webinar for CURE, which is on innate immunity and how we can study it in the laboratory. Although I said to Nikos, Nikos suggested the topic, and of course I said immediately yes. Then by looking at it, I found that it was a bit more complicated than I originally thought. So I tried to structure the presentation to the following 4 topics. So to first of all, talk to you about some of the basic concepts of innate immunity, because at least first of all, half of the participants maybe are not exactly from the field. And second, remind you about the basic ways innate immunity functions, that it uses receptors, receptor systems that broadly recognize pathogens, and then talk to you about the models, assays, and technologies that can be used to study innate immune processes, and talk a bit towards the end about systems approaches and why this may be the way forward. So as you all know, as most of you at least know, there are several barriers to invasion of a microbe or a pathogen. And first of all, there are non-specific physical and chemical defenses, and that includes barriers like the skin, mucous membrane, stomach, acid, lysozyme, tears, and so on. And also there are also expulsion mechanisms like the coughing, the sneezing, the vomiting, diarrhea, which work to move away any potential pathogens. Then there is innate immunity, which is part of the immune system, and I'll talk to you more about it, but it's the natural immune defense system that our organs possesses. And then there is adaptive immunity, which is acquired, which is specific for specific pathogens, and it involves cellular and humoral immune responses. Now, there are some very fundamental differences between innate and adaptive immunity. And first of all, an innate immune response is an immediate response. It is initiated within seconds, and that's what is something very important you have to have in mind. It targets groups of pathogens rather than specific pathogens, and it has no memory. So in principle, it should be the same every time our body encounters a pathogen. Then there are adaptive immune responses, and these are a gradual response generated over days, over 3-4 days, I would say, for T-cell activation. It is specific for specific pathogens, so it targets specific pathogens, so it develops for specific pathogens, and it also has a memory, so it remembers. So if we have a re-exposure to a pathogen, then our immune system responds faster and better. Now, in terms of the cells of adaptive immunity, these are the lymphocytes, so the T cells, the T lymphocytes, and the B cells or B lymphocytes. T cells have specific receptors called T cell receptors for pathogens, and that's how they get the specificity to specific pathogens. And B cells have the B cell receptor or membrane-bound antibody and antibodies. So they are the specific arms of the immune response. All the remaining cells can be considered to be part of the innate immune response. But the most prominent ones that we traditionally view as the main innate immune cells are the macrophages and the granulocytes. Dendritic cells are also part of the innate immune response, but they are important, as I'm going to tell you in a bit, for bridging the innate and adaptive immune responses. Now, as you understand, it's very important for the immune system to be balanced. If there is underactivation of the immune system, then that makes us susceptible to infections or cancer development. And if there is overactivation, that leads to allergic reactions, allergic diseases, like allergic asthma, but also autoimmune diseases. So the immune system has to be balanced, and the innate immune system, the innate immune response is key for achieving that. Now, going to the receptor systems, the principles that innate immunity uses, that involves pattern recognition receptors. So the most important aspect of innate immunity is how it is activated, and that determines how exactly the innate immune system will respond. So for a long time, there was a lot of effort to understand and identify the pattern recognition receptors for decades or centuries, maybe we can say, till the late 90s. Before I go to the actual receptors, so what these pattern recognition receptors do is that they activate conserved structures, microbial structures, and as we know now, also some non-microbial structures. And that includes, just to give you a few examples, that includes, for example, from bacteria, from gram-negative bacteria, LPS, beta-glucan, flagellin, bacterial DNA, which is unmethylated or undermethylated, and also structures from gram-positive bacteria, such as lipoproteins, lipoteichoic acid, unmethylated DNA again. And you can think of phages as well, that phages are made in bacteria, so they contain unmethylated DNA or RNA that is different from the mammalian. For our own RNA, it contains proteins that are synthesized in bacteria, so it contains structures that are potentially recognized by pattern recognition receptors. Of course, we know nothing about these and how exactly they can be recognized. Now, as I started telling you before, for a long time, the pattern recognition receptors, the main pattern recognition receptors, have been elusive, but the whole field changed from the late 90s onwards, that it was shown that the Drosophila protein, initially identified in Drosophila for its role in dorsal-ventral polarity during embryogenesis, was also key for mediating antifungal immunity in the fruit fly. And this was a seminal work published in Cell 1996, and this work and that work that follows gave the result in the Nobel Prize in these two groups involved. So 2 years later, in mice, it was in mammals, it was showing that TLR4, by analogy to the Drosophila TOR, was the first toll-like receptor identified to be a pattern recognition receptor and recognize LPS, the bacterial lipopolysaccharide. And the way these experiments were done was by looking in mice, where everyone knew that there are some strains of mice, a specific subtype of C57 black 6 mice, and some C3H mice that do not respond well to LPS, that are hyperresponsive to LPS. So they did classical positional cloning and went on and identified TLR4 as the gene involved for this phenotype. After the whole field searched, and there was recognition and identification of many pattern recognition receptors, including TLR2 and many others, as the human genome and another animal genome started coming out. Now, according to the structure, toll-like receptors have a specific localization in the cell. So toll-like receptors that recognize structures that are found extracellularly, like LPS, flagellin, or various bacterial cell wall components or yeast wall components, and these are recognized at the cell membrane, like TLR4, 5, 1, 2, 6, and 10. Whereas there are toll-like receptors like 3, 7, 8, and 9 that recognize bacterial or viral nucleic acids and are therefore found in the endosome. So they recognize bacterial and viral structures in the endosomal compartment. Now, another important aspect that is key when we study and we're trying to understand how innate immunity is activated and what it does has to do with the localization, the expression pattern of toll-like receptors. And the initial concepts were that toll-like receptors, well, first of all, toll-like receptors are found abundantly in immune cells, and that makes sense. Not all immune cells do express the same panel of receptors, and this is an older generic slide, but macrophages overall express most or all toll-like receptors known, Dendritic cells also express most toll-like receptors known. Again, there are dendritic cell subsets that express different toll-like receptors than others. Lymphocytes express fewer receptors, and even the ones shown here, some of these have been challenged whether they are really functional in these cells. Granulocytes express a good pattern of receptors, so overall immune cells express well toll-like receptors. And the original idea was that the toll-like receptors are found in immune cells, however that proved to be wrong, and now we know that epithelial cell surfaces, other stromal cells, non-immune cells overall in the body do also express the receptors. Now, what about the structure of toll-like receptors? Toll-like receptors have a very conserved structure that you will find in all toll-like receptors, and that makes them part of the family. So they have an intracell and transmembrane domain, and an extracellular portion that is very characteristic of toll-like receptors, and it contains leucine-rich repeat domains, which are amino acid stretches of containing leucines with this motif. And this is very important for toll-like receptors because this is the one that is involved in the recognition of microbial structures. So this is critical. I'll come back to that. And then there is an intracellular domain called T domain or toll interleukin-1 receptor domain, which is also shared, has homology with IL-1 family members, and this is very important for signaling. So once early on, when we knew which were the we had some information for the first toll-like receptors, it was easy to go to the human genome data as they were coming out and look for molecules that have a transmembrane domain, a leucine-rich repeat, and a T domain, and that was the way by which all the other toll-like receptors were identified. Now, coming back to the recognition part, this is complex, and you need to know that it's not just the leucine-rich repeat domain depicted here with purple that is important, but there's a whole complex that is assembled. And in the case of LPS, that includes LPS binding protein, which is replaced in the serum and binds to LPS, MD2, also a co-receptor CD14, which lacks an intracellular or has a very short intracellular portion and does not signal, which is also part of the LPS recognition complex. The fact that toll-like receptors contain a T domain also made it easy to identify, to unwind the first signaling events induced by toll-like receptors. And these, by analogy to IL-1, involve adapter molecules that contain a T domain as well, but lack a transmembrane domain, so they are cytosolic, like MID88. Now they involve MAL, TRAP, and TRIF as well, TRIF and TRAM as well. So there is a complex formed in the intracellular part of the receptor that involves MID88, the other adapters that I just mentioned, IRAC family molecules, that this leads to the recruitment of traps and eventually the activation of I-kappa-B kinases, or IKKs, and NF-kappa-B, and gene expression patterns. So this is a main, and this is very important when I will be talking to you for things we look at and how we studied innate immune processes in the laboratory. So this is a very important, this is the central pathway that all toll-like receptors activate. And that leads to the activation of NF-kappa-B and inflammatory responses. However, this is the whole picture is much more complex. First of all, some toll-like receptors have a second major arm that is activated by LPS, that is also activated. TLR4 activates this process that leads to the activation of another family of transcription factors termed IRFs, or interferon regulated transcription factors like RF3, RF7, RF5, RF1, and so on. And these lead to the expression of another set of genes called interferon-stimulated genes, and including interferon as well. And this also seems to involve partly NF-kappa-B, a different response, and different NF-kappa-B subunits. So there is complexity. And in addition to this, this second part, I have to tell you that, in addition to LPS, is also shared with TLR3, TLR7, 8, and 9, so the toll-like receptors that recognize nucleic acids. Now, in addition to these, are multiple other signaling cascades involving the activation of MAP kinases, PI3 kinases, and other transcription factors that are partly understood, but there is complexity, and I'll come back to that a bit later, briefly. So, which are the functional consequences of innate immunity? So, first of all, the functional consequences, of TLR activation. First is induction of innate immunity, and that leads to cell activation of the main immune cells of innate immunity, like macrophages and neutrophils, activation of antibacterial responses, and activation of antiviral responses. So the transcriptional programs that I mentioned, that include at the center NF-kappa transcription factors and IRF transcription factors, and limited upregulation of inflammatory cytokines, but other responses as well. Now, one of the key components that I mentioned is the induction of interferons. These are very early induced genes, and I just want to remind you here that these are activated as early in follow the infection, for example, in this case of a virus, and you can see here with blue the viral load, and you can see how rapidly type 1 interferons are induced. The adaptive immune response follows. I'll comment on that later on again. So the other functional consequence of TLR activation is the instruction of adaptive immunity. So toll-like receptors and innate immunity create the environment in which the adaptive immune response will develop, and the type of the adaptive immune response that develops. And that depends a lot on the cytokine and chemokine profile that toll-like receptors, pattern recognition receptors, innate immunity in general activates, induces, and then also it involves more specific arm of this process, which involves the interaction of the antigen-presenting cell, the dendritic cell, with the T cell, and that determines the interaction, the co-stimulatory molecules, the status, the presence, the expression, the intensity of the signal, the duration of the signal, and so on, but also the cytokines produced, determine whether T helper response of a type 1 or 2 or TH17 response or a regulatory T cell response will develop. Together, the dendritic cells determine not only the T cell response activated, but also the B cell response, the production of antibodies. And they provide, they activate a key part of that is the activation of the T cell response, which provides a help to B cells, so B cell help for antibody production, antibody affinity maturation, and so on. So this was my introduction for toll-like receptors. For the sake of completeness, I have to also talk about some of the other toll-like pattern recognition receptors. Innate immunity is not just toll-like receptors, but it involves another very important group, also termed inflammasomes, and also another important groups of cytosolic sensors outside the endosomes, which includes MDA5 and RIG-I. And again, these recognize nucleic acids. So the inflammasomes are a big category of pattern recognition receptors. They are complexes that are formed, and they contain something very characteristic. They contain the involvement of node-like receptors, or NLRs. There are many NLRs as well. Again, the most known are NLRP3, NLRP2, 9, 1, 10, node 1 and 2. These involve a CAR domain, in node domain, the intracellular receptors, and then, very interesting, they include a leucine-rich repeat domain, which by analogy to toll-like receptors, and what I told you, this is very important for ligand binding. So the same leucine-rich repeat domain seem to be very characteristic for pattern recognition, not only of toll-like receptors, but also node-like receptors and inflammasome assemblies. Now, in terms of activation of the inflammasomes, this is more complex. This involves the formation of multimeric complexes. Some of the components of multimeric complexes have to be induced. It depends on the cell type, and this can be induced by toll-like receptors or inflammatory cytokines like TNF. And even the expression of NLRs, like NLRP3, is induced. So it requires signal 1, which means the induction of inflammation components. And part of the inflammation component is also the inactive form of alpha-1 beta, pro-alpha-1 beta. And then there's the signal 2, which is the recognition and the activation of the inflammation complex. And this shows you here the example of NLRP3 inflammation, and which its activation upon recognition requires the results in the assembly of this multimeric complex that involves, in the case of NLRP3, in the case of the NLRP3 inflammation, NLRP3 ASC, another adaptive molecule of the inflammasomes, which contains also parent domains and CAR domains, and procaspases. The most prominent is procaspase 1 in the case of an NLRP3 inflammasome. This is the one involved, and that becomes activated upon always ligand binding, so upon pattern recognition, and that becomes activated and cleaves to alpha-1 beta to alpha-1 beta. And it can also cleave alpha-N18, which is another alpha-1 family member, to pro-alpha-18 to IL-18. These are released, and these activate further the innate immune response. So the inflammation is a very interesting part of the pattern recognition systems of the organism that specifically activates alpha-1 family members, which act as alarms of the immune system as well. For the sake of completeness, as I told you, there are also cytosolic sensors, very important, I think, for cure, for everyone should be aware of these. These include RIG-I, MDA5, LGP2. RIG-I is very important because it activates viral RNA, and it drives the expression of interferon related gene programs, including type 1 and type 3 interferons. So this is a very important cytosolic sensors shown here. So this is, I think, most of the introduction I wanted to make about toll-like receptors, about pattern recognition receptors overall. I just want to put this in a bit of a perspective, and of course, as you understand, depending on the ligands that a microbe carries, this shows you here the example of Salmonella typhimurium. So if it has ligands for TLR2, TLR9, 2, and 5, or whatever, so this determines also the type of immune response that will become activated. So you can have a response A, a response B, a response C, and so on, depending on the pathogen-associated molecular structures that each pathogen or microbe possesses. Also, that includes the other pattern recognition receptors. So if there are also ligands for RIG-I, for the cytosolic sensors or inflammation, that again influences and adds to the complexity of the immune response that is going to develop. Now, going to the second part of my talk, that has to do with how we can study innate immunity in the lab. And that is very complicated because it depends on exactly the system and the model we want to use and we want to study. And this has to be done ad hoc. So I'll give you some of the basic principles and assays and that we use in the lab and how these can be adapted for specific questions that you may want to address and that we also want to address. So this is an analysis that has to be systems that have to be used ad hoc. Now, how can we assess innate immunity in vitro? First of all, we can monitor the molecular signaling events that are activated by pattern recognition receptors. And that reiterates some of the previous slides that I had. And again, it's a very simplistic slide of the signaling cascade that all like receptors activate. And someone can study that in the lab. by looking which of these molecules, which of these pathways are activated, whether something, a ligand, a compound, an inhibitor, the presence of other cells, if we're talking about co-cultures, the influence of a factor we have identified, FH, a component of FH, a component of a microbe, how this can affect classical innate immune activation. So the first part that someone can look at is the signaling cascade that leads to the activation also of transcription factors. The simplest system, the simplest and most standard systems used involve studies in monocytes and macrophages. These, in the case of human systems, come from peripheral blood mostly. So monocytes are studied either in the context of peripheral blood mononuclear cells or in isolation as monocytes. These can be differentiated into macrophages, or they can be studied in culture as monocytes, although in culture they rapidly start differentiating into macrophages. That's the reality. And they can be put in a petri dish, 96 well plate, a 24 plate, or whatever someone is interested at, and in medium, and then they can be activated with TLR ligands, cytokines, compounds, inhibitors, as I mentioned, other cells. In the case of mouse cells, we can also use wild type of knockout cells that lack specific genes, and we can then address the function of these genes in culture. In mouse systems, we prefer to get monocyte macrophages for bone marrow, where we differentiate monocytic progenitors and macrophage progenitors from the bone marrow into macrophages. But we can also get them from the spleen, from other major organs, or we can even activate, in the case of the lung and the respiratory tract, macrophages of the respiratory tract. These we put them in culture, and then we can culture them with the stimuli I mentioned, and get the cells, get extracts, and look at intracellular signaling cascades. And this is done most by Western blotting. There are other ways that they can be done these days as well, including some kits, ELISA-based kits that can look at the presence of phosphorylated mediators, or even Luminex kits that can look at this. But the classical, traditional, and more common, and with a wider then the system that where we can look in a wider way for most intracellular signaling cascades is done by Western blotting. So we can look. These are just examples I took from paper. You can look at the activation of JANK kinase, P38 MAP kinase, activation of I-kappa-B kinases, phosphorylation of kappa-B alpha, activation of interferon stimulator regulated transcription factors like IRF3, and you can also look at the nucleus, and you can look at the presence of transcription factors in the nucleus, or you can do electrophoretic mobility shift assays. This is from one of my very first work and publications, and this shows NF-kappa-B activation in response to LPS, TNF, cytokines, and this is our more specific electrophoretic mobility shift assays, or you can see by Western blot I-kappa-B alpha. But electrophoretic mobility shift assays are assays where you can label the binding sequence for specific transcription factors with phosphorus P32, and then you can incubate these with the extras you have and run them in a gel. And this, if there is NF-kappa binding, that appears, as you can see here, as complexes. This is a free probe. It ends up at the bottom of the of the gel, because it's free. However, when the probe binds to its target, its transcription factor, in this case NF-kappa-B, the probe is delayed and appears delayed here, and that shows NF-kappa-B DNA complexes. This includes the various complexes. NF-kappa-B includes 5 subunits, so they're from different homo-heterodimers and so on. So studying intercellular signaling cascades is very important when we want to understand how the very basic innate immunity is regulated and how it can be affected, how it can be modulated from the factor of the system that we want to address. So this is something that can be used. It's simple. It requires a bit of expertise, but not too much, and it's easily adaptable to the in various ways to the questions that someone wants to address. Now, the second thing that someone does using the same system of macrophages is to look, okay, this was a signaling part, but does that affect inflammatory mediators like cytokines, chemokines, but also other molecules that are secreted like MLPs that are also very important in an inflammatory response. So in the cultures that I just showed you, here we can collect supernatants, we can which can be examined then for the expression of cytokines. First of all, by ELISA, if you have enough supernatant and if you repeat sufficiently the experiment, you can do as many ELISAs as you want. And that's also the expression of some of the cytokine in response to a TLR2 ligand, another TLR2 ligand, TLR3 ligand, a modification of the TLR3 ligand. These also hit inflammasomes, LPS, a TLR4 ligand, RF48 that is recognized by TLR7 and 8, and CPG that is recognized by TLR9. So you can see that the response is differs as well, that most of these, I can't remember what the color show, but most of these induce TNF. However, it's only poly-IC, the TLR3 ligand, and the TLR7/8 ligand, and the bit CPG that induce in these cells interferon alpha or interferon beta. So if, for example, we have a component from phages, if we have a component or components or a set of components or microbes that are relevant to cure, we can use this, we can add this in culture, and we can look at the cytokine profile, and we can get an idea from the cytokine profile of how innate immunity is affected. We can look, first of all, at these components independently. If on their own they can activate innate immunity, but we can also look if the presence affects the way the innate immune system responds. So if many of these pattern recognition receptor ligands act. And of course, we can go back to signaling processes and address how this can be affected at the molecular part. And I insist on that, as I believe, that just by looking for cytokines is a bit very generic, and we want and we need in the context of cure, but overall in the field of innate immunity, to seek mechanistic evidence for observations, and that includes molecular events. Now, of course, the technology is developing, and Luminex is the way, is a much more expensive, but much more holistic way to look at cytokines. And you can look with Luminex at 20, 25, 30, 50 cytokines together, with very little need for material. So this is something we can look at in the context of cell cultures, but also in the context of any fluid that may be available from patients, and where we want to look at cytokine expression and chemokine expression in these patients. Of course, we can also look at even cytokine gene expression and general activation of cells by flow cytometry. We can use intracellular flow cytometry to look at the cells that express, for example, TNF, and this shows you an example here that shows macrophages, monocytes expressing it in culture, but also T cells expressing it. This is with the stimulus PMA, a bit artificial, that activates well T cells, but in most of the cases, it's monocytes that make most or the vast majority of TNF in an innate immune response. Well, in the body overall, not just in the immune response. Then we can also use flow cytometry to look at activation of immune cells, such as macrophages as well. And you can see here expression of CD8 and how this can be upregulated in response to stimuli. So this is a histogram plot. We can look at that by dot plots or many other ways by flow cytometry. So we can get macrophages, look at the intercellular signaling cascades, so get molecular evidence for that, and also get protein expression like cytokines, chemokines, which are key regulators of the immune system overall. We can look at this in the supernatants. Now, when we talk about innate immunity, of course, we think of phagocytosis, of microbes, and microbial killing. And this is again can be done by simple assays in culture, where again we can look at macrophages. Many of what I'm talking about, of course, are applicable for dendritic cells and neutrophils as well. Similar types of assays can be used. In the case of macrophages, we can label bacteria and green, and we can look by fluorescence microscopy or focal microscopy. If we want better pictures, we can look at the uptake of microbes by macrophages or granulocytes, actually shown in this case, neutrophils. This is an example. So this is an example of on the left of neutrophils, and this is an example with macrophages in culture, where we can add in the media label bacteria, and you can see that macrophages take up some label bacteria, but this is very much enhanced when these macrophages become activated by TLR activation. This shows CpG here, the TLR9 ligand, and you can see here also effects of at the bottom on peptidoglycan as well. And this shows you can measure the bacteria that these cells take. So if you have 1 bacterium per cell, on average, this you can get a graph. If you have 2 per cell, 3, and so on, you can enumerate this. And as you can see, TLR ligands increase the number of bacteria that the cell takes to 4, 5, or even 6 in the case of CpG. And this is an effect that's interesting, that it can be inhibited if you inhibit P38 kinase with this classical SB inhibitor here. So same setup in terms of culture, or very similar setup in terms of culture, which can be used to look at the bacterial uptake. And if you wait long enough, you see also bacteria killing, the bacteria disappearing. Another important assay that I could not omit has to do with a different aspect of innate immunity. I talked a lot about the activation of the innate immune response and how important that is to activate inflammation. However, innate immunity is also very important for shaping its resolution, for determining its resolution. And one of the key aspects of innate immunity and of innate immune cells, like macrophages, is to take up apoptotic cells. And so ensuring that cells that have done the job, immune cells, inflammatory cells that have done the job, are properly cleared from the tissue. So they are properly removed in a non-inflammatory way. And this can be done, it's an interesting assay. The way we do these things is we get, again, the cells we want to study for phagocytosis. That can be classically macrophages, they can be dendritic cells, they can be, again, neutrophils. And we also get target cells, the cells that we want to see how well these are cleared. And we label these target cells, and of course we make them apoptotic, usually by UV radiation or other chemicals if needed. We label these cells and we add them in culture to the, in this case, macrophage culture, and we can see then how well these cells are taken up by macrophages. Again, we can look at signaling cascades that are activated. These are examples from the paper, irrelevant, but it's, the basic concept is that we can look at signaling cascades by getting back macrophages and looking at the signaling cascades there. We can look at the phagocytosis process, the uptake of apoptotic cells, and we can do that again, as we have labeled the cells. We can look by fluorescence microscopy how many of the cells are taken up. We can quantify them by flow cytometry as well. And you can see here with red apoptotic cells, with the green the untreated cells, and you can see that these cells are taken up in various degrees depending on the treatments and the lack or presence of activators or inhibitors, or the lack of presence of genes. So this is, again, can be done in culture. Now, an aspect that always comes to our mind is whether we can characterize different states of innate immunity, and the way we do in the adaptive immune system, the way we do for adaptive immune response. And we know that in adaptive responses, we can say this in allergic response, we can say Th2-driven allergic response, we can say this is Th1-driven response, this is Th17 or neutrophilic response. However, for innate immunity, we cannot really easily discriminate different states of innate immunity. Because this is every time ad hoc, it depends on the specifics, on the specific system, the specific infection, specific timing, and so on and so on. Maybe it's too dynamic in response to be categorized, and the fact that it doesn't, it's broad and it doesn't have memory, it doesn't stay eventually, makes this really difficult. But one of the few states that we can recognize innate immunity involves the states of M1 and M2 macrophages. So we can discriminate macrophages, so the principal, the central cell of innate immunity, we can discriminate in it and characterize in it different states that we call M1 or M2. In reality, these are a spectrum of states, but we can broadly call them M1-like or M2-like macrophages. And M1-like macrophages and M1-like innate immune responses are the ones that we classically view as pro-inflammatory, the ones that make iNOS, key for killing bacteria, the ones that make TNF, alpha beta, and this is indeed, as I just said, key for pathogen clearance. And we can also view M2-like macrophages, which can be, depending again on the context, more regulatory, they express agenase, they express IL-10. These macrophages are more potent in taking up apoptotic cells and mediate nephrocytosis, that I just mentioned to you. They are important in taking up debris. So they are more important as an inflammatory response and as an innate inflammatory response resolves. So we can characterize these two different states, and if we expose, say, an undifferentiated macrophage into various pathogens, we can look and in a way categorize the innate immune response into an M1-like or M2-like immune response. We tend to see M2-like macrophages as well in allergic states, maybe due to the presence of high levels of IL-10 and IL-13 there. So this is an important aspect for you to remember in your studies. And a final aspect I want to mention about the in vitro culture systems is something that is coming up. It's early days. It's mostly shown in, would say, and clearly shown in experimental animal models and some cell culture systems. So evidence really in the human organism remains to be seen. But that involves the training of innate immunity. And there is emerging evidence that although we say, and that's why I want to present that one of the basic, the dogma in innate immunity is that innate immunity is the same. It doesn't have memory, it doesn't change. The innate immune system responds the same way every time we get exposed to the same pathogen. So, however, it's becoming apparent that innate immunity can also be altered depending on the exposures we have from the very early life and later on as well. So it can be trained, it is trained all the time. And that seems to involve, of course, key to innate immunity training are macrophages, as these are more long-lived, and dendritic cells, as these are more long-lived cells in the organism. Training seems to involve epigenetic reprogramming of these cells. So, as I said to you, you can have training of these macrophages to be long-lived and more M2-like or more M1-like or have more specific states. And that determines and they can also be trained also to the pathogens, not just in states, but also to the pathogens. So the classical examples involve the study of C. albicans. fungus, but macrophages, the bottom line is that macrophages can respond better if they get, the same macrophages can respond better if they get re-exposed to C. albicans compared to macrophages that have never been exposed to C. albicans. And the same holds for some of the bacterial pathogens as well. So there is training that is not clearly understood at the moment, but there is a lot of research going on. So there is training of macrophages, and this in terms of cure. And this training, I forgot to tell you, that can be affected also by vaccination, secondary infection, and secondary exposure, and so on. In terms of cure, this is very important because what could affect training of innate immunity more than a microbiome, or than the microbiome? What could affect more training of innate immunity, the gut or the respiratory tract or the skin, than the microbiome that we possess, and also alterations in the microbiome that could be present in healthy individuals, but also in patients. So this is a very important aspect of training of innate immunity. The way they do it at the moment, mostly experimentally, culture, is we get macrophages, like in the systems that I presented to you in culture. We expose these to the agent that we want to study, and then we can look at the program of these macrophages in culture, how this changes. The good thing about macrophages is that they are relatively longer lived or long-lived, but they're not too long-lived either. So there is also a difficulty there, how to look at long-lived macrophages, which in culture they stay, but they don't stay for too long. So you can't keep them for a week, you can keep them for 2 weeks. But so there is some internal difficulty in terms of experimental, the way someone can approach that experimentally. In animals, that may be easier because you can administer the components you want, and you can look in a more longer term. In humans, evidence for that can only come in types of cohorts, like the Q cohort that has been designed, and again, this is has lots of hurdles that need to be overcome, and it involves again lots of associations and some indirect evidence. But these are things for sure that we're trying to address in cure. Now going to the final part. This involves how we can study innate immunity in vivo. And the way people do it is really in experimental animals. Now, there are numerous infection models that can be used, and innate immunity is studied there. You understand that this is an unlimited number, but that includes for sure viral infection systems, bacterial infection systems, where we can administer, someone can administer Staphylococcus aureus, Pseudomonas aeruginosa, Listeria, or other bacteria, lots to be administered, but these are the more classical ones that are studied in culture. Or fungal infections like Aspergillus, Candida albicans that I mentioned, and so on. So innate immunity is studied in the context of, specifically in the context of the model that someone is looking at. A simple way to look at innate immunity in vivo is to measure cytokines in the plasma and serum. So that involves administration of the ligand or the pathogen-associated molecular structure or the agent that we want to study, or even a microbe, and look at in vivo at the expression of cytokines that are made. That has to be done quickly because these cytokines are rapidly upregulated in a sepsis model, for example, and downregulated. So that requires early time points to look at, for example, TNF or interferons, and later time points that someone can look at IL-10, IL-6, and chemokines, and so on as well. So in vivo, this is the classical kinetics that someone would expect. This is in plasma, and TNF peaks within an hour. It's a very rapidly produced cytokine of, for example, LPA stimulation. IL-1 beta follows, and IL-6 follows later on, starts coming up and stays up for longer time points. Actually, IL-10 follows and so on. So a rapid test for innate immune response in vivo is through the administration of pattern recognition receptor ligands, like TLR ligands, like LPS, or the pathogens themselves. And then get serum or plasma and measure the inflammatory molecules produced. Cytokines being the most prominent ones. Now, in terms of bacterial infection models, again, I said it, that depends on the model that can be used. And this shows an example, one of the models that are used, depending, again, on the question that someone wants to address, is Staphylococcus aureus. But again, depending on the question, that's what I wanted to show you here. There can be inoculation of the pathogen intravenously or intraperitoneally, that is systemically, subcutaneously, or intranasal, that we are mostly interested in our studies and in our field. That can lead, if administered intranasally, that leads to lung inflammation, tremendous lung inflammation, pneumonia, as you can see here. And there are various ways that this can be assessed by looking histopathologically, by looking as well at the doing morphometry in the lung and getting histological scores, looking at the presence of inflammatory cell infiltrates in the bowel, looking at bowel function, looking at cytokines produced there, looking at bacterial load. That's something very important as well. So there are various assays to look at the extent of an infection and how the infection progresses and how innate immunity can affect this. In a setting of viral infection, this is from the system we use in our studies. You can also look at the virus. This shows H1N1 influenza virus. You can use various doses of the virus and look at body weight loss, survival of the mice, at increasing dose of the virus. Of course, survival worsens here. And you can look at the level of the tissue, the infiltration of immune cells, the damage. If you look at high magnification of the lung, you can assess lung function by using lung mechanics, looking at viral load, and look at the innate immune response by mostly processing bowel and lung tissue, and look at the production of all the innate inflammatory cytokines, the innate immune cascades, and how this can affect the response. Now, as you understand, it's very innate immunity does not act on its own, but in all the systems that I described to you, adaptive immunity also gets activated. And of course, if you look very early on, you look at the innate immune response, the production of interferons, you can look at the activation of NK cells as well, or innate lymphocytes, and other key aspects of innate immunity that are activated. However, there is also adaptive immunity that follows. And it's difficult to discriminate between innate and adaptive immune responses, and because also adaptive immune responses also come and feed back to the innate immune response. And the adaptive immune responses affect the innate immune response, they affect macrophage function, they affect the way macrophages take up phagocytose, kill microbes, they affect the way they respond to a second silence of microbes. So it's very difficult to, it's a cycle. So it's very difficult to discriminate the innate immunity components. And the way we do that in experimental animals is we use rag knockout mice, which lack both mature T and B lymphocytes. And in that way, you can look at the innate immunity, purely at the effects of the innate immune response in an infection, for example. As you can see here in the example of influenza virus, mice that lack the adaptive immune part, shown here with purple and red, are much more susceptible into different doses, are much more susceptible than their wild-type counterparts. However, there are there are descriptions and there are pathogens that are mostly handled by innate immune responses, and the adaptive immune system is less important. Influenza virus infections is not one of them, and generally respiratory viral infections are not in this category. And now, as you understand, there is complexity, and this, I see it's four, these are a couple of my final slides. Where the field is going, it's this to understand innate immunity in the context of also of adaptive immunity, and try to understand how these two interact and how innate immunity develops, can be affected by, at the very, very early stages, by pathogen structures or microbial structures that may not be pathogenic, or other exposures that we get exposed, other environmental exposures or insults. This requires systems biology approaches, so requires combinations of genomics, proteomics, metabolomics, animal models, human models, human genetics, and so on. And a very interesting way, and you can look at that, that this was done in a seminal study published in Cell a few years ago. This involved the use of different, these are some of the schematics of different doses of viruses, and the study of different doses of influenza viruses, different types and strains of viruses, some lethal, some sublethal, different influenza viruses, and a very omics, holistic analysis of lung patterns, including transcriptional patterns, cytokine patterns, but all in unprocessed lung, or at the level of the whole lung, but also at the level of the individual cells. So this involved a lot of sorting of cells, of neutrophils, of macrophages, T cells, of other immune cells as well. And this looked at the identification of signatures that could discriminate a lethal from sublethal innate immune response. So it's a very interesting, I mean, to cut a long story short, it's a very interesting systems biology approach. I think the best that I've seen that you can look at. But this ended up discriminating signatures like that were protective against influenza infection, and that included the activation of antiviral signature involving what we call interferon-stimulated genes, or IA genes, and it also involved the discrimination of the signal of molecular signatures that do damage and cause lethality in the end of the day, and cause ARDS in humans and acute lung injury. And that the signatures that were more related to led to acute lung injury, involved extensive neutrophil activation, excessive production of cytokines like TNF and IL-1 beta, but not others, and of course leading to vascular pulmonary damage and lethality. So systems biology approaches are the way forward. Of course, it's easier to be said than done. It's very complicated. It's early days. It's computational power, so on. But that's where eventually this is going, and this will be eventually very helpful for discriminating the innate responses from the adaptive, the way they interact, the way they get breathed, and so on. So to conclude, I want to start with this: that innate immunity is an integral part of the immune response of the immune system, and it cannot be viewed really solely as an independent process. And if it is studied, it has always to be in context with the adaptive immune system. Some characterization of innate immunity can be achieved through the use of cell culture systems and assays, and this can be done in monocyte, macrophage, and neutrophil cultures, where we can look at signaling, molecular events, cytokine chemical production, other inflammatory molecules that may be important, phagocytosis, microbial killing, and so on, but also immunomodulatory functions. Innate immunity can also be studied in vivo, but this is model and context dependent, very model dependent. It requires ad hoc analysis. I use many times the word ad hoc because this is how it is. And that new technologies and approaches have the potential to change that, and enable the study of innate immunity at the systems level. And I'm going to finish by reminding you that the Nobel Prize in Medicine in 2011 that was given for the recognition of pattern recognition receptors for Jules Hoffman for the toll discovery in Drosophila, Bruce Butler for the LPTLR4 discovery in mammals. And then this was not given just for immunity, but it was given also with the discovery of the dendritic cells to Ralph Steinman. And the dendritic cells are the cells that bridge innate with adaptive immunity, and that shows what I just said to you, that innate immunity cannot really be viewed in isolation. But this is, innate immunity is important for the early protection, but because it also instructs adaptive immunity. And that recognition of the importance of these early events of innate immunity for the adaptive part also were recognized with the Nobel Prize Award at the same time to Ralph Steinman. And I'm going to stop here, and I'm open to discussion and comments and questions that you may have. Thank you.